Hi everyone, we are going to be reviewing Unit 5 Heart Disease. This is for the PLTW, Project Lead the Way, PBS course. We're going to be looking at the structure of the heart as well as some complications that are associated with things such as high blood pressure and high cholesterol. First thing that we're going to be looking at is the structure, specifically the blood vessels. The first blood vessel that we're going to look at is the arteries. Arteries are going to transport blood away from the heart. Typically, arteries are going to have oxygenated blood. Now, there is one exception. That's going to be the pulmonary artery. The pulmonary artery transports deoxygenated blood to the lungs. The next blood vessel is going to be the veins. Veins bring blood into the heart. Again, this is going to be typically deoxygenated. This, though, also has one exception, and that's going to be the pulmonary veins. Pulmonary veins are transporting blood from the lungs back to the heart, so they do have oxygen in them. Our next thing that we're going to be looking at is actually the structure of the heart. You should be familiar with the different parts. I do think one thing that will help you is drawing that heart box that we did. Remember, when you're labeling the sides of the heart, it's not as though you're looking at it, it's as though it's inside of you. On the left-hand side, then, we have the right atrium. Remember, the atrium is like a welcoming area, so that's going to bring the blood in. Underneath that, we have the ventricle. On the other side, we have the left atrium and the left ventricle. The other thing is that you want to draw that figure 8 that's showing how the blood is being pumped from the lungs back to the heart, to the body, back to the heart. The right side brings it to the lungs. From the lungs, it goes to the left side. The left ventricle, remember, it's very muscular. It's going to be capable of pumping that blood all around the body. After it goes to the body, it goes back to that right side. Part of this is asking you to trace the flow of blood, blue being deoxygenated and then red being oxygenated. So if we start off first with our blue color, remember blue is going to represent the fact that it's deoxygenated. Deoxygenated is going to be on the right-hand side. So looking here, we have the superior vena cava as well as the inferior. Blood goes into the heart, through the valves, then into the ventricle on the right-hand side, and then it goes to the pulmonary artery. Note the pulmonary artery is deoxygenated. Remember, that's one of those exceptions. The next example is going to be when there's oxygenated blood. Oxygenated blood is brought to the heart from the pulmonary veins. Note again, pulmonary veins are going to be an exception because they have oxygenated blood. It then goes through the left atrium, through the left ventricle, and then it's going to go to the aorta. From there, it's able to go to the rest of the body. Over here, we have a couple of valves. These are going to be the 4 valves that you want to be familiar with. Again, my advice would be really just kind of utilize that heart box that we've been talking about. Here I have my right atria, right ventricle, left atria, left ventricle. You're going to want to know which valve is in between each one of those structures. So first up, we have the tricuspid valve, which is going to be this here. It's called tri because it has 3 structures associated with it. Over here, we have the pulmonary valve. That pulmonary valve actually takes it to the pulmonary artery. On the other side, we have the mitral valve. Note the mitral valve is in between the left atrium and ventricle. And then finally, we have this aortic valve. That aortic valve is going to be in between the left ventricle and the aorta. A little bit more in terms of heart structure. Number 4 says identify the structures that prevent backflow. Remember, we want to make sure that that blood is moving in the correct direction. You don't want oxygenated, deoxygenated, right? A little bit of backflow happening there. That would be bad. What is going to prevent that are going to be the valves. Next up, we have that term pulmonary. A lot of times we hear pulmonary associated with that artery and that vein. that's going to be responsible for transporting blood between the heart and the lungs. Pulmonary means lungs. Identify the chamber that has the thickest muscular wall. Again, thinking back to that heart box, and I really suggest that you draw that heart box when you're trying to answer these questions because it is going to assist you. You want to think which one is pumping it all to the body. That's going to have to be the most muscular. The one that's actually transporting it to the lungs, that blood doesn't have as far to go. The part that's going to be the most muscular is going to be the left ventricle. And again, that's because it's going to the entire body. Identify the structure that carries blood to the lungs. The one that's going to do it to the lungs is going to be the pulmonary artery. Remember, arteries are going to go and transport blood away from the heart. Also remember that this is one of those exceptions, so therefore usually arteries have oxygenated blood, but in this case it has deoxygenated blood. Over here, we're doing a little bit of an analysis. Again, remember, you want to cross off the ones that you know are definitely not correct. It says an ultrasound reveals a hole in the septum. The septum is the area that's going to separate the left and the right-hand side of the heart, as shown in the figure. What are the impacts? One says the respiration rate decreases. Well, here what's going to happen on this side, you have deoxygenated blood, and on this side, you have oxygenated blood. That blood is mixing. Therefore, your body is not going to be getting enough oxygen. And because of that, your respiration rate is actually going to increase. So we're going to cross that one off. Number 2 says blood vessels in the hand and feet dilate. That's not going to be correct. Number 3, the heart works harder to provide oxygen to the tissues. That is correct because your brain is sending a signal saying, hey, body parts are not getting enough oxygen. So the heart's going to pump faster and faster and faster in order to get adequate supply of oxygen to them. Oxygenated and deoxygenated blood will mix. That's what we just saw in that picture, so that's definitely true. And then the last one says blood flows back from the pulmonary artery into the right ventricle. Here we have the pulmonary artery right here. And the thing that prevents the backflow is actually the valve. So the septum is not related to that. What are some technologies that we are going to be able to use? We used all of these in class. Note what you're going to use for blood pressure, the heart rate. We use those heart rate sensors, the heart electrical impulses. We use the EKG, lung and heart sounds. We use the stethoscope. First thing that we examined in these experiments was going to be the heart rate. Remember, heart rate is just going to indicate your heart beating. The rate at which your heart is beating could indicate that potentially you might have a medical problem. Some things that can affect heart rate that we tested in particular were how could you lower heart rate? You can lower heart rate by resting, laying down, sitting. Your heart is not working as hard. So first thing when you wake up in the morning, your heart rate most likely is going to be the slowest that it's going to be all day because you've been resting and laying down. What can make for a faster heart rate? That can be things such as standing, exercising. Certain types of medications can increase your heart rate. A big thing that you're going to have to be able to do is evaluate whether or not a patient has high blood pressure or if they have low blood pressure or if they are normal. So you'll be given a patient and you have to make a determination based on the numbers that you see in front of you. Blood pressure, remember, is indicating the force exerted on the walls of the blood vessels while the ventricles are doing what? So there's two types of blood pressure. If you remember, there's going to be a top number and a low number. The top number is top number and a bottom number. The top number represents systolic pressure. Remember when we were taking the blood pressure and we had the cuff on, what we were listening for when we were looking for the systolic number was a sound. That sound is indicating that that heart is going to be contracting. Right? Ba-bum, 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 ba-bum. That is systolic. Ideally, what you are looking for is a value that's going to be less than 120. That's going to be considered healthy. So a value of 120, right, greater than 120 mmHg indicates a high reading. Next up, we have diastolic. Diastolic is suddenly when that sound stopped. So systolic, you heard ba-bum, 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 ba-bum. There was a pause, and then you might have heard another beat, a pause, another beat. That's your diastolic. This is going to be when those ventricles are relaxing. Once again, we're looking for specific values in order to consider someone healthy or if someone's at risk for heart disease. A value that is going to be greater than that 80 mmHg indicates a high reading. Looking here, this is giving you what the different ranges are. Normal is 90 to 120 for systolic. Diastolic is going to be 60 to 80. Please make note of these pre-hypertensive, hypertensive 1, hypertensive 2. You will be evaluating someone and having to make a determination how high their blood pressure is, and then also what type of treatment you're going to need to be giving them. What can a person do in order to lower their blood pressure? A lot of times we just keep talking about lifestyle modifications. Obviously, if you're pre-hypertensive, so your blood pressure is going to be slightly elevated, right above that 120 over 80. These are things that you can do in your everyday life to lower it without the use of medication. So first up, we always have lifestyle. And as with all the lifestyle changes, what we're really talking about is going to be lose weight. We're going to modify our diet. And when we're talking about modifying diet, a lot of times we're talking about lowering your sodium intake, lowering fat intake. Then we also have exercise. Now, if you try those things, or if your blood pressure is very high, you most likely will have to go on a blood pressure medication. The medication for this is going to be an ACE inhibitor. Now, something that was a little bit challenging is going to be the next thing that we look at. That's going to be how you actually read an EKG. There's a lot involved in the EKG. You're going to have to know how to label some of the parts. You will also have to know what each wave that we looked at is trying to tell you in terms of heart working properly or not working properly. Here we have an EKG. Again, EKG is going to look at that electrical impulse that's happening inside of your heart. Over here, I have labeled the major nodes that you need to know. Probably one of the more important ones is going to be the SA node. I always think the SA node is the start. Many times I call the SA node the pacemaker. So if someone does get a pacemaker, that's to replace their SA node because most likely it's not firing properly. And if it does not fire properly, that means your heart is not going to be beating properly. So SA node, again, that's going to be your start or the pacemaker. The next section that we have is going to be the AV node. I'm just going to draw the line a little bit over here. The AV, what you're going to notice is actually between, right, kind of nestled in there in between the right atrium and the right ventricle. So AV, that's what it's standing for, atrium ventricle. What that is going to be responsible for is a pause. Now, why do you want to pause? You want to pause because that SA node is going to contract the atria. You want to make sure that all the blood that was initially in that atria is going to get into that ventricle before that valve closes. That AV node allows for all that blood to be pumped from the atria down to the ventricle. Let's atria fully empty. Now the other parts are the bundle of His, bundle branches, and the Purkinje fibers. Right here, this one, that is going to be bundle of His. One's down here. We've got branches. Then over here, these are the Purkinje fibers. Even if one of those nodes is going to malfunction, the next one will take over for it. The problem is that your heartbeat will not be as efficient at pumping the blood. That's why people get pacemakers. if their SA node is misfiring. Now, how do we actually go about reading these EKGs? So there's a lot involved here. First up, we've got PQRST. That's what this picture is showing right there. Notice it goes in alphabetical order, and what you're really looking for is just that first initial spike. That's going to be your P. There's going to be a dip, which is your Q. Big spike, which is the R, and then S and T follow that. What the P represents is going to be when the atria contract. Now remember, instead of contract, sometimes they're going to call that depolarize. Remember, the atria are the top part of the heart. The QRS interval is going to be when your ventricles contract. And again, instead of contract, you could use that word depolarize. Your T wave is going to be when your ventricles, sorry, that's spelled wrong, relax. Now, when we say relax, another word that you can use is repolarize. Finally, we have this R to R. That's telling you what your heart or your pulse rate is. And if I'm looking at this one, I've got my R, R. What you're looking for is the space in between those to remain consistent. If that space remains consistent, that is going to indicate that your heart rate is consistent. Based on those EKGs, you're supposed to be able to diagnose different types of disorders, or you're supposed to be able to look at them and tell, hey, there's no P wave, there's no ST wave. You need to be able to make that determination, so it's really important, even if you need to sketch out for yourself what that PQRST looks like. First step for one problem that we specifically looked at was premature ventricular contractions. We're going to look at both the rhythm of this, and if you go back to your notes, you actually have examples of this in there. When you have PVCs, what happens is that your rhythm is irregular. So if you're looking at those R to R values, they are going to change. They're not going to stay consistent. And that's because the ventricle is more or less contracting too early, right? That's what premature means. So that ventricle contracts too early, and then you're going to have a miss of the P, and because of that, those two R's are most likely going to be closer together, and it's going to be inconsistent. The other thing is that the P wave is missing. The P wave represents those atria contracting. What happens is that in premature ventricular contraction, the atria don't contract. All of a sudden, the ventricles contract before the atria get a chance. So the P wave here is absent. Next up, we have ventricular tachycardia. Tachycardia means that your heart is beating fast. The rhythm itself is regular though, so R to R, R to R. That stays consistent. It's just a very, very quick beating that's happening. Also notice it says ventricular. So the ventricle's pumping really, really fast, and because of that, it skips over the atrium. So once again, that P wave is going to be missing. So for this, we have the rhythm is going to be regular. Again, that means R to R is regular, but the P wave is missing or absent. And the last thing, actually, one of the last things we're going to look at is there are going to be two things that we're looking at. One thing is HDL, called high density lipoprotein, and the other one is LDL, which is low density lipoprotein. Technically, you need both of these. Many times we associate one with being good and one with being bad, but both of them are necessary. HDL is going to be your quote-unquote good cholesterol. You would like that value to be greater than 40. Another thing to note is what the actual function is. So this brings cholesterol to the liver. When it brings that cholesterol to the liver, that is going to make sure that it's not going and building up inside your blood vessels. If it builds up inside your blood vessels, that's going to increase your risk of heart disease. Next up, we have the LDL. LDL is quote unquote bad cholesterol. You want this value to be less than 100. What's the problem with this? This one brings cholesterol to your cells. Now, Your cells, they need cholesterol in order to function properly. The cell membrane, for example, is made up of cholesterol, so you need that. But let's say you have an excessive amount of cholesterol in your body, what winds up happening is you get a buildup in arteries. When you have a buildup of cholesterol inside your arteries, that is going to increase your risk of heart disease. Other things to note, there's another component kind of when we're talking about heart and artery health, and that's going to be your triglycerides. Triglycerides are fat that store energy from food. If your levels are too high, you're going to increase your risk of atherosclerosis, which remember that is going to be the hardening of the blood vessels. A normal value would be below 150. High levels are going to be anything above 200. So once again, you're going to get a patient, you're going to have to determine what their cholesterol level indicates about them, and then also what type of treatment you're going to want to go and implement for that person. So we have total cholesterol. Ideally, your total cholesterol level would be under 200 milligrams per deciliter. Next up, we have your LDL. Remember, this is the one that you want to make sure is kept low in check underneath 100. So we have less than 100. That is going to be the one that is responsible for cholesterol building up inside of your arteries. You get a hardening of arteries, then you wind up having higher risk of heart disease, heart attack, things such as that. HDL, you want to have over 40 milligrams per deciliter. Because again, that's going to reduce your risk, because if there is cholesterol inside of the blood, that's going to bring it back to the liver for storage instead of it building up inside of those blood vessels. We have 3 different patients and we need to decide if they're healthy, if they have high cholesterol, or maybe they're borderline high and could just do some sort of lifestyle change. First up, we have 185. 185, this value less than 200, and because of that, this person is going to be healthy. Total cholesterol for the next person is 215. 215 is obviously above 200, but the thing we want to keep in mind is that this is really borderline high. Yes, it is definitely higher than the recommended number, but probably your first steps here would be lifestyle changes. If you cannot modify your cholesterol, right, you don't get any sort of decrease, and you've tried exercise, diet, losing weight, none of those things have worked, then you might have to go on medication. After that, total cholesterol 285 for our third patient, that is going to be high. Once again, lifestyle is always going to be important, so you're going to try to get your patient to make lifestyle changes. But at this value, most likely you're also going to want to implement a medication. The type of medication that you would take for this is going to be a statin. The last part of this unit is really going to be to evaluate the gel electrophoresis. This is the one that we actually ran through the real gel electrophoresis chamber, and we need to identify if someone has a specific type of allele or not. From that, we're going to make a pedigree. You need to know what the difference is between a male and a female, how we're going to represent the carrier, how we're going to represent, a recessive trait, a dominant trait, things such as that. Here on the left, they give you an introduction as to the type of disease, what it is, who has it, those types of things. So here we have the disease. It's called, here we have the disease. It's called FMH. FMH is a autosomal recessive. Remember, recessive means that you need 2 little alleles, right? 2 small letters. So it's going to be masked or covered up by that dominant allele. Moving down to the bottom, it also is telling us that the female patient and the male patient, they do not have this disease. Now, just because they don't have the disease does not mean that they are not a carrier for the disease. It goes on to say that they did testing on a brother 1 and brother 2, and we have to figure out what their genotype is. Remember, a genotype is referring to the genetic makeup. And they give us 2 controls. The first one is non FMH, meaning that person does not have the allele. If they don't have the allele, that means they have the dominant allele. And because of that, that's going to be our big F. If they have the FMF allele, they are going to have the recessive allele. So that's going to be a little f. Looking here, right, anyone who has those two lines, they're going to have at least one big F. The next thing that you have to do is you got to figure out the FMF allele, which is going to be represented by that little h. Sorry, little f. From this, you should be able to tell the genotypes of all of the people listed, the female brother 1 and brother 2. So looking here, patient female. If we're looking, she definitely has a big F. But if you go up, you'll also notice she has that blue line as well. That's indicating that she has a little f. So the female patient is big F, little f. She's a carrier, or sometimes they're going to call that heterozygous. Right? Hetero means two different alleles, so she has a big F and a little f. Brother 1 only has that blue band. If he only has that blue band, that means he has two little f's. They're not going to show up as separate bands, and that's because when the gel electrophoresis runs, all of the genes, right, the genetic marker band, is going to be in that same location. So those two little f's are literally right on top of each other on the gel. And then the last person is brother 2. What you'll notice is brother 2 only has those yellow lines. Since he only has those yellow lines, that's going to be big F, big F. From this information, we are going to have to go and actually make a pedigree. Looking at the pedigree, just to remind us, we had female 1, 2. Female was a carrier. 1 was, he had the disease. And then the other one was healthy. He was not a carrier, and he did not have the characteristic, the recessive allele. So here we have our different generations. We have the mom and the dad listed. Note the mom and dad are both carriers. If we are looking at the second generation, if we start off, this is going to be my female. The female was a carrier. We're going to go shade in half of that. Next up, we have brother 1. Brother 1, he had the disease, so he's totally shaded in. And then finally, we have brother 2. Brother 2 did not have the disease. Note that the circle is representing the female and the square is representing the male. Also note that the carrier is half shaded in, whereas the one with the recessive trait is totally shaded in. And then finally, the one who does not have that allele at all, they only have the healthy alleles, they're not shaded in. Note there is this other sister. She's not tested, so we don't know what she is. The next question, though, goes on to kind of ask that. Which genotypes are possible for the sister? Looking back really quick, note that the parents are both carriers. So from that, you're going to have to go and make a Punnett square. Remember, you separate the alleles when you make a Punnett square, and then you drop them down and bring them across. So looking here, she can be homozygous dominant, meaning that she has the two dominant alleles. She could be heterozygous, meaning she has one dominant and one recessive. Or she'd be a homozygous recessive, meaning that she has those two recessive alleles. The male patient's father has FMF and the male patient's mother does not. What is the correct genotype for the male patient? I know this was a little bit confusing, but if you go back to that initial reading that we had, it actually states here that the male is healthy. He's healthy, but that doesn't mean he's not a carrier. Because remember, when you're a carrier, you're not expressing those traits or showing those traits. So we have to think, it says that the male patient father has it. That means his dad is little f, little f. If his dad is little f, that means the only sperm that he was capable of making was a little f. It said on the top, though, that the The male doesn't have it. That means that his mom, her egg had to give a big F. Because of that, his genotype, genetic makeup for the male patient is going to be big F, little f. Last little section here, what's the probability of a couple's future child having this? Remember the female patient was a carrier, and then the male patient is a carrier. So you have big F crossed with, big F, little f, crossed with big F, little f. That's the same Punnett square as was up here. So the chance that their child is going to have this is going to be 25%. Because again, they are both going to have that recessive allele that they can pass on to their child, and there's a 25% chance that their child will get that. The last thing that we're looking at is going to be what you can do for someone in order to reduce their risk of heart attack. This is really just like vocabulary terms. First, medical term for a heart attack, that's going to be an infarction. So we have myocardial infarction. What do we call it when you have chest pain? That's going to be angina. When you use dye or x-rays to look at the heart, that is going to be an angiogram. Now, let's say the EKG came back and you were concerned about it. You would go through and look at the angiogram. The tiny balloon that's put inside of someone is called an angioplasty. Remember, the angioplasty can be used in order to clear small blockages. When they go and create a whole alternative path, that is going to be coronary bypass. Now, you want to do a coronary bypass if there's a large blockage. You know, if there's a 90% blockage, sometimes they'll call that an occlusion. If there's a 90% blockage, you want to make sure that you go around that blockage, because most likely an angioplasty or a stent is not going to work long term. So the bypass is your best bet there. Last one is tiny balloon with a permanent metal device. That is going to be a stent. And again, that also includes the angioplasty. They use the balloon in order to blow up the stent and lock it in place. And then they deflate the balloon and take that out. The last thing is kind of, if you have every single thing that's going to heighten your risk of getting heart disease, that's called metabolic syndrome. All these topics we talked about, if you're diabetic, if you have high blood pressure, if you have high triglycerides, high cholesterol, and if you're obese, you're just increasing your risk of having heart disease, stroke, heart attack, all those things. That's called a metabolic syndrome. Remember, metabolism is talking about all chemical reactions that occur in your side of your body, inside of your cell. And if you have this metabolic syndrome, you're more at risk for dying, right, or having a condition such as heart disease.